Hello everyone, welcome back to the class. In our previous class we discussed about synaptonemal complex which is a protein structure which is responsible for forming the pairing between homologous chromosomes during meiosis, especially during zygote, which mediates chromosome pairing. So, synaptonemal complex is a tripartite proteinaceous structure which is consisting of two parallel lateral elements and a central element. Each lateral element is associated with a pair of sister chromatids, and the central region which is present between two lateral elements is composed of numerous transverse filaments. In vertebrates, the lateral elements are made up of protein SYCP2 and SYCP3, whereas central element is made up of proteins like SYCE1, SYCE2 and SYCE3. So the main function of synaptonemal complex is stabilization of homolog pairing and promoting the formation of crossing overs. And we also discussed the third substage, that is pectitin. After synapses is completed, the cells are said to have entered to the pectitin stage of prophase, where they remain for many days. At this stage, there is a large recombination nodules present in which a large protein complexes are present, which contains RAD51. In eukaryotic version, it is REC A, that is recombination A protein. So there is a major process happening here, that is crossing over. Crossing over is a reciprocal exchange of equal and corresponding segments between non-sister chromatids in the pair of homologous chromosome. I mentioned that some of the cells will remain at this stage, at this pectitin stage, for days. Why? Because it is waiting for another homologous pair. For example, if we consider in human beings, so if this is paternal and if this is maternal, the maternal chromosome has to be present so that crossing over can occur. Without this maternal or without this paternal chromosome, there is no crossing over. So the chromosomes remain, or the cells remain in pectitin stage for several days, waiting for the interval where the other parental chromosome comes and starts the crossing over. So this crossing over leads to recombination of genes present on two homologous chromosomes. So the crossing over, evidence for crossing over is found by Stern working with Drosophila, and H.B. Creighton and B. McClintock working on maize. So it occurs in a tetrad bivalent state of the pectitin stage, although it is said that throughout the chromosome length, anywhere chromosomal crossing over can happen, but there are some regions which are having crossing over frequency high. So the regions where the crossing over frequencies is high is known as hotspots, and the regions which are having lower crossing over frequency, that is cold spots. This chances of crossing over is more in hot spot when compared to cold spot. So here in pectitin stage, make note that synapses is completed and crossing over is happening. So next stage is Diplotene stage. In Diplotene stage, what happens is, in Diplotene stage, first there is desynapsis. In Diplotene stage, there is desynapsis, where the intimate pairing which had happened in zygote will remove or will separate. So the desynapsis begins. Okay, the synaptonemal complex will disassemble, or we can say it dissolves. It dissolves. It allows to homologous chromosomes free. It allows to homologous chromosomes free, and it is pulled away from each other to some extent. But consider now, recombination has already happened. See, this is one homologous chromosome, maternal and paternal. So now, there is crossing over happening here, or consider happened already. Now the synaptonemal complex is dissolved. Synaptonemal complex is dissolved, and each bivalent is remained joined by two or more chiasmata. So what do I mean about this statement? Chiasmata is the representing site. It is site of or site where the crossing over has occurred. So here synaptonemal complex is dissolved, which allow the two homologous chromosomes in a bivalent to pull away from each other. So in oocytes, or we can say developing eggs, so this diplotene can last for months or years, since at this stage that the chromosome decondenses. In this stage the chromosome decondenses and they engage in RNA synthesis. Why RNA synthesis is done or is happening here? To provide storage materials for the egg, right? In the extreme conditions, the diplotene chromosome can become highly active in RNA synthesis and expand to an enormous extent. So it produces lamprey chromosomes found in amphibians and some other organisms. Next is diakinesis. So diakinesis is the last stage of prophase I, of meiosis I. So diplotene merges imperceptibly into diakinesis. So there is no perfect demarcation between diplotene and diakinesis. So the stage of transition to metaphases, or the stage for stage is prepared for metaphase after this process. So in this diakinesis, mainly RNA synthesis is ceasing and chromosomes becoming condensed, thicken and become attached from the nuclear envelope. So each bivalent contains now 4 separate chromatids, with each pair of sister chromatids linked with their centromeres. After this has done, the cell enters into metaphase I and followed by anaphase I. See here the stages, which gives the summary or outlook of the overall prophase I. Here in leptotene, the centromere is prominent and the tips of the chromosomal ends are attached to the inner nuclear envelope, forming a bouquet. So therefore the leptotene stage is known as bouquet stage. The chromosomes are thin here. Zygotene, where the synapses or intimate pairing is happening between two homologous chromosomes. So here there is intimate pairing with one major protein structure, that is synaptonemal complex. It is a zipper like structure which collects or which organizes or which pairs the homologs of same homologous chromosomes. In pectin stage there is a crossing over. So where you can see crossing over, it is a reciprocal exchange of equal and corresponding segment between non-sister chromatids. Okay. Next, after pectin, Diplotene stage is there, where you can see the chiasmata. The chiasmata is representing the sites at which crossing over has occurred. After that, in diakinesis, so the RNA synthesis is ceased, chromosomes become more condensed and it becomes more thickened. It detaches from the nuclear envelope. After that, what? Metaphase I, where in metaphase I, so there is it is marked by some events happening which will be discussed in the next class. Thank you.